Thank you for the introduction. I'm glad to be here and would like to share our recent data on using focused ultrasound to stimulate the multiple somatosensory areas of the human brain. I would like to, this study is presented on behalf of the principal investigators, Drs. Yonghan Jung and Seung-Sik Yu. So in the last FOSS symposium in 2014, we presented that various types of tactile sensations, such as tingling or heaviness, were elicited by FOSS stimulation of the human primary somatosensory cortex. We were curious whether we can elicit the operator-defined types of tactile sensations using the FOSS stimulation, perhaps by stimulating the additional somatosensory cortices, such as S2. Our objectives are, first, to examine the types of tactile sensations elicited by FOSS stimulation on the separate somatosensory cortices in humans, targeting the S1 and S2 of the right hand. Second, to test a hypothesis on eliciting intended types of tactile sensations, such as vibrotactile, pressure, warmth, and coolness, by stimulating the S1 and S2 using FOSS. We also wanted to examine the feasibility on simultaneous stimulation of the multiple brain areas and the safety profile. This is our study design. A total of 10 healthy volunteers participated. In pre-screening procedure, fMRI CT scanning was performed to be used for later image guidance for targeting the S1 and S2. The multiple FOSS sessions were conducted with different sonication conditions, and the post-sonication safety profile also examined with a 2-month follow-up. Using functional MRI, we were able to identify the allocated functional areas of the S1 and S2 to the external tactile stimuli of vibrotactile, pressure, warmth, and coolness given on the palm of the right hand. So this slide shows the schematic of the FOSS setup and the image guidance procedure for targeting the S1 and S2. We used 2 sets of single-element 210 kHz FOSS transducers. This slide shows the sonication parameters based on our previous study. And FOSS was given at 35 watts per square centimeter ISPPA with a 50% duty cycle. The size of the focus measured at full width half maximum was 60 millimeter in diameter and 38 millimeter in length. And we would like to emphasize that the actual site of neuromodulation may occur at much higher maximum, like 90% of the maximum. This illustrates the locations of the elicited sensations. Overall, these results are similar with our previous study results of the S1 stimulation. And new findings are that the sensations can be elicited by simultaneously stimulating the S1 and S2, or by stimulating the S2 alone. These are the types of the elicited sensations. Vibrotactile, pressure, warmth, and coolness were reported, while the types of the tactile sensations did not match to the intended types of sensations. However, 2 individuals reported matching types of sensations during the simultaneous stimulation of the S1 and S2, or the stimulation of the S2 alone. So we applied the instant intensity of 35 watts per square centimeter ISPPA, and the post-FOSS safety profile of 2-month follow-up revealed no abnormal findings across all subjects. So in discussion, we demonstrate that transcranial FOSS to the human S1 and S2 elicited various tactile sensations. Most types of the sensations were similar to those from our previous study on the S1 stimulation. The elicitation of the warmth and vibrotactile were new findings. Stimulation of the S2 alone also induced the creation of tactile sensations. Using FOSS, we also showed the possibility of simultaneous stimulation of the S1, S2, which has not been feasible with TMS or tcUS. However, further caution is needed to make sure that there's no crosstalk between the multiple sonication. These are our collaborators and funding sources. Thank you very much.